I am Snigdha Bharadwaj, Assistant Professor, ITS College of Pharmacy. I welcome you all in my session, and in this session we will be continuing discussing about reaction kinetics. Basically, we will focus on its pharmaceutical application in this lecture. So, let us get started. So, in our earlier sessions, we have discussion about reaction kinetics and rate of reaction rate laws, their mathematical expressions, and in this particular we are going to discuss about pharmaceutical applications of reaction kinetics, why it is important to understand these type of reaction orders, rate laws, why it is essential to study these. So, basically chemical reactions such as decomposition of medicinal compounds, processing of drug absorption, distribution and elimination from the body, shelf life determination. So, while talking about reaction kinetics and identifying their basic parameters, so it also affects the drug product and its stability, right. So, they are very helpful in identifying the shelf life of the drug and drug product, and pharmaceutical products. Basically, it has impact on the drug processes like absorption, distribution, elimination from the body. We can establish a correlation between the in vitro and in vivo data using these reaction kinetics, and the chemical reactions such as decomposition, the pattern how the drug is getting degraded during the storage period. So, we can also identify using these reaction kinetics. So, order of reaction, basically why it is important to know, manner in which the rate of reaction varies with the concentration of the reactant. So, most processes involving ADME, that is absorption, distribution, metabolism and excretion, can be treated as first order processes. So, in our last session we have discussed what is zero order reaction. That means the rate constant is not getting changed with respect to concentration of a reactant participating in the reaction. First order reaction means the ADME basically treated as first order reaction, where it is the rate of reaction is getting changed with the concentration of the reactant participating in the reaction. Some drug degradation processes can be treated as either first or zero order processes. So, that is why we need to identify the order of reaction to track its degradative pattern of a drug compound. Some drug substances obey Michaelis-Menten kinetic process, that is non-linear kinetics. So, at this point of time the drug when we correlate biopharmaceutics and pharmacokinetic parameters with the drug performance. So, generally we say that drug is following linear kinetics or non-linear. So, generally Michaelis-Menten equation or kinetic process is basically represent the non-linear kinetics of drug that is following the process. So, determination of order of reaction, how it is getting determined. So, use of rate reaction, the data collected in kinetic reaction should be substituted into the integrated form of equation of various order. In our previous session of the rate of reaction, rate laws, identifying different kind of equations. So, we have discussed also about the integrated values of first order, zero order and second order reactions. So, the process under test should be considered to be that order where the calculated k is a rate constant, remains constant within limits of experimental error. So, generally during identifying the rate of order of reaction, we keep k as the constant value to minimize the experimental orders. So, half life method for a zero or pseudo first order reaction, T half is proportional to initial concentration of reactant. So, basically T half or half life of a drug or substance or a reactant in a particular concentration, the time taking to reduce its half concentration to the initial value is termed as T half. So, half life method for a zero order or pseudo order or first order reaction, T half is proportional to initial concentration of reactant. So, T half for a first order is independent of CO. So, generally radioactive decay usually follows the first order reaction kinetics. Graphical method for a zero order or pseudo first order reaction, plot of C versus time. In our earlier discussions, we have discussed that this type of graph concentration and T value. So, this basically the graphical methods generally we can plot for zero order, first order, second order and pseudo orders as well. So, it is a linear representation, the slope for first order plot log CO minus CT and T is linear. So, sometimes in zero order we get this type of slope, and for first order this type of slope. So, different types of slope we can get in the graphical method when we put our experimental observation and represent it in the form of graphical methods. Apparent zero order reaction kinetics. So, suspensions are a special case of zero order kinetics in which the concentration of drug in solution depends on its solubility. So, if we talk about the formulation, that is suspension, a special case zero order kinetics are followed in which the concentration of drug in solution depend on its solubility. As the drug gets solubilized within the system, it will give the value of concentration. So, the concentration depends on the solubility of the drug product within the system. As the drug in solution decomposes, more of it released from a reservoir. So, basically when we talk about suspension, we are saying that suspension is a system which consists of two immiscible phases, one is solid and other one is liquid, and we form a system using some sort of suspending agents. So, basically the drug is not miscible with the solvent. So, this we can say that this is a reservoir type of particle suspended all over the solvent. So, this is a special case that a drug gets solubilized within the system in by means of any solubilizer or any suspending agent. So, the drug in solution decomposes, more of it is released from a reservoir of suspended particles. So, these are suspended particles we can say, and a reservoir of a suspended particle. So, we call it reservoir of suspended particles. They are not solubilized at this point of time, but over the time they may get solubilized within the system. So, however, the concentration reaching to its solubilized form in the system will thereby making the concentration in solution constant. So, that is why in this particular suspension case zero order kinetics are usually followed. The effective concentration is the drug equilibrium solubility in the solvent of formulation at a given temperature. So, the other one is the solvent, the how much amount is going into solubilized form with the solvent at a given temperature, generally analyzing in zero order reaction kinetics. So, temperature and the rate constants, this is the another correlation, temperature with respect to the rate constant. So, temperature has a dramatic effect on reaction rate. For many reaction, an increase of 10 degrees centigrade will double or triple the rate. So, factor affecting rate of reaction, in that particular part we have discussed the effect of temperature. So, there also we have discussed this specification that if the temperature is increasing by 10 degrees centigrade, the rate of reaction will be double or triple at that particular temperature as compared to normal one. So, experimental data shows that K increases exponentially as the T increases. So, directly proportional, temperature is directly proportional to the rate of reaction. This is expressed in terms of Arrhenius equation. So, Arrhenius equation, the K is a rate constant A e to the power minus E A divided by R T, whereas K is rate constant, A is frequency factor and E A is activation energy. Activation energy basically it is increasing with increase the temperature. So, higher the T, larger the K and the increased rate of reaction of the reaction. Now, activation energy. In order to be effective, collisions between particles must exceed a certain energy threshold. In collision theory, we discussed that higher the collision, the particle must collide in order to react. So, if the activation energy will be higher, the collision will be more prominent as compared to the normal temperature, and it will produce the desired rate of reaction above its threshold, right. So, when particles collide effectively, they reach an activated state, right. So, activated state, the energy difference between the reactants and the activated state is the activation energy E A. So, when we say that activation energy, we are saying that particles in a system or in a reactions are participating with each other. More the reactions, the activation energy will be higher. So, number of collisions basically represent for each reaction will give the higher activation energy or rate of reaction. So, the lower the activation energy, the faster the rate of reaction. So, basically the energy difference between the reactants and the activated state, energy difference. So, at the time of the between the reactants, the energy of the reactant participate in a particular reaction and the activated state by increasing the temperature or solvent or pH or moisture or any kind of approach we are or catalyst. So, we are trying to increase the rate of reaction. So, the lower the activation energy, the faster the rate of reaction. So, smaller E A values generally give larger F values, rate constant higher and increased rate of reaction. So, this is the inverse relationship of activation energy and the rate of reaction. So, in order to be effective, collision between particle must exceed energy threshold. So, they reach an activated states. Temperature and collision energy. So, increase in temperature causes an increase in kinetic energy of the particles, that why more frequent collisions are expected. So, in this, so the temperature rise may result in the higher collision among the particles participating in the reaction. So, collision and reaction rate increases. Right? At higher temperature, the fraction of collisions with sufficient energy equals to or greater than E A increases. So, at higher temperature, the fraction of collisions basically with sufficient energy is equal to or greater than E A. This E A is your activation energy, right. So, increases the reaction rate, therefore, increases. Higher the collision, greater the rate of reaction. The effect of E A and T on the fraction F of the collision with sufficient energy to allow reaction. So, basically this is a table representing the temperature value, E A activation energy, it is a threshold energy threshold, above that it is the particles are exceeding. So, at 50 kilojoule per mole, F is the fraction of collisions. So, collisions basically how much collisions are taking place. So, at T 298 K. So, the collisions will be 1.7 into 10 to the power minus 9 at 75 kilojoule. So, the collisions will be higher. As you see the relationship at lower temperature, the collisions are lower as compared to this value. So, and it is gradually increases. So, how it is calculated? The temperature F at E A is equal to 50 kilojoule per mole. So, in terms of centigrade, so at 25 degree centigrade, the value will be like same. 35 it is 3.29 and 45 it is 6.12. So, we see that the relationship the temperature is getting increased, then the fraction of collisions with sufficient energy is also increased. So, we can say that the rate of reaction is increasing with the increase in temperature. Calculating activation energy, how we can calculate? So, E A can be calculated from the Arrhenius equation. How? This is the basic Arrhenius equation. we have discussed in earlier sessions also. So, integrated form will give you this type of representation, straight line form. So, if the data is available at two different temperatures, if we have different temperatures and we are saying that activation energy is also impacting with the effect of temperature. So, integrated form will be represent, this is for general straight line representation for activation energy identification. So, integrated form, two temperatures or two constant will be there. So, E A will be like 1 divided by T 2 minus 1 by T 1. So, this is how we represent at two temperatures activation energy or different temperature values. So, graphical determination of activation energy will be like integrated K rate constant, integrated A, and this is basically slope activation. So, this is how we can calculate E A using this graphical methods time. So, the formula will be like integrated K integrated A minus E A R 1 by T. So, this is how we graphically represent the activation energy, and we can identify its value using these graphical methods. Now, after activation energy, transition state theory. So, an effective collision between particle leads to formation of transition state or activated complex. So, basically if no collision, as per collision theory, we must have heard that if no collision, no reaction will occur, right, between particles or reactants or reactant particles. So, transition state theory says an effective collision between particle leads to the formation of transition state or activated complex, means we can say if partition collides, they are changing to its final product, right. The transition state is unstable. We generally say in an equation that A plus B will give you C. So, particle of A and B must collide in order to give the C. So, maybe some intermediates may form during this transition. So, generally we say it that transition state or activated complex. So, transition state is unstable and C is a final product. So, in between there may be chances to form intermediates. So, transition state is basically unstable species that contains partial bonds, right. It is transitional species pathway between reactants and the products. So, we in simple term we say A plus B will give you C, C is the product, A and B are reactants of the reaction, and during this process intermediates may form, right. Some intermediates may form and these are called as unstable species and contains partial bonds. So, they may take part in a reaction and they may form in between, but at the end you will get the desired product, right, C. The transition state exists at the point of maximum potential and energy. So, this state which are producing unstable species are known as transition state, and it occurs at the maximum potential energy. The energy required to form transition state is the activation energy. So, using this activation energy, reactants collide with each other and form different unstable species and to give the desired final product. So, this energy required to form this transition state, basically activation energy is required. So, we have different reaction mechanism. So, the mechanism of a reaction is the sequence of single reaction step that make up the overall equation. The mechanism of a reaction is a single reaction step that make overall equation. So, how the drug is basically using different sort of substrates or reactant, we are finding or getting the final product. So, they must have some sort of mechanism. So, basically single step says about the reaction, how the reaction is taking place. The individual step of the reaction mechanisms are called elementary steps. So, in some cases we must have read that A plus B will give you C plus D and after adding some other substrate will give D plus E or E plus F. So, multi-steps equations are there. So, each individual step will be called as elementary step. So, because each one describes a single molecular event, each elementary step is characterized by its molecularity, the number of particles involved in the reaction, the number of particles of the reactant involved in reaction. So, the rate law for an elementary steps can be deduced from the reaction stoichiometry, reaction order equal to molecularity of the elementary step only. So, we generally find the rate of reaction or reaction orders for elementary step of a reaction. So, elementary means the reactants molecule participating with each other. So, we generally identify the reaction order for a single step, and for every step we need to identify the reaction reaction orders for different elementary steps involved in the equation or chemical reaction. Molecular structure and reaction rate. For a collision between particles to be effective, it must have both sufficient energy and the appropriate relative orientation between the reactive particles. So, for collision between particles to be effective, there must be effective collision to give the desired product, right, desired product. So, they must have sufficient energy as well as the appropriate configuration for the interaction, right, reacting particles. The term A in Arrhenius equation is the frequency factor. This is the frequency, how much time it is reacting with the other particle of the reactant, right. So, it can be calculated like K A E to the power minus A. This is the Arrhenius equation. A is P Z, P is orientation, the exact configuration through which it is getting interacted, and this collision frequency, how much time the particle is colliding with the another one. The P is specific for each reaction, that is orientation, the active form of the drug or the most reactive form of the particle taking place in the reaction, and is related to the structural complexity of the reactants, that the L form is active more than the D form. So, this is how enantiomer is more effective than the normal one. So, depending on its structural composition, the P value gives its orientation probability factor, and collision Z is the collision frequency. So, that is how we identify A using this Arrhenius equation, frequency factor. Rate laws for general elementary steps. So, elementary steps you see here. A, elementary means single reaction we are taking. A, product, unimolecular, molecularity is unimolecular product we get, and rate will be A. So, if we are producing two different components using reactions. So, the elementary step will be 2, product bimolecular, and the rate will be expressed in terms of K A to the power 2. A plus B product bimolecular. In a particular reaction we may have different products altogether, and this is also the molecularity is bimolecular, because we are getting two different molecules from a reaction, and the rate will be expressed in terms of K A B. And 2 A plus B product termolecular, that means 3 molecules particularly we are getting from a reaction. So, the rate will be like K A 2 B, because here the 2 molecules of A, the concentration of A will be higher as compared to B. So, correlating mechanism with the rate law, a valid mechanism must meet 3 criteria. Elementary steps must add up to the overall balanced equation, the equation should be balanced, and it must be reasonable and must correlate with the observed rate law. So, for it must follow the physical laws for the rate of reactions. A mechanism is a hypothesis, we can't prove it, but we can't prove it is correct, but it is consistent with the data to establish that the particular reaction is following this type of mechanism and giving this appropriate products at the end. So, we can basically take it as a hypothesis, and it is a useful method for model for the reactions. Catalysis basically it is speeding up a reaction, a catalyst which enhance the rate of reaction without getting involved in chemical reaction or losing its structural complexity while in the reaction. So, in general it provides an alternative reaction pathway that has a lower total activation energy than the uncatalyzed reaction. So, in that the lower activation energy, that means the rate of reaction will be higher. A catalyst will speed up both the forward and reverse reactions and does not affect either the overall yield of a reaction. Right. Shelf life determination, basically we use these reaction. So, decomposition or in determining the shelf life, how the drug shelf life or drug product shelf life, for how much time it is like stable when we keep it in store or during the storage time. So, we can also identify these shelf life of a particular product using these reaction orders and kinetics. So, basically different formulation additives and its effect on the formulation, through this we can identify its decomposition factors. The stability projection for shelf life. So, using this equation we can identify the stability Arrhenius, the log value or log equation of Arrhenius K by 2. So, basically it is a two constant two component reactions. So, 10% of the drug to degrade with 90% of intact remaining is based on Arrhenius. So, shelf life, basically this using this equation we can determine shelf life. Chemical instability basically represent it is a loss of potency, accumulation of toxic degradative products. So, when we say that drug is chemically unstable, we are saying that it is not in its active form and it is totally degradative. So, degradative, either loss of potency, accumulation of toxic degradatives, right, excipients degradation, color change, or these are the signs of chemical instability. Solid state versus solution stability, chemical reaction proceed more rapidly in liquid state and as compared to solids. So, serious stability problems are more common encountered in liquid medicines. Order of dosage form, like stability is like solution has lesser stability as compared to suspension and then tablets. So, tablets are more stabilized as compared to liquid form. These are references. I hope you understand the basic pharmaceutical applications with all relevant significance of each parameters like shelf life, half life, rate constant, Arrhenius equation, effect of temperature and reaction orders. So, these are all basically reaction kinetics important in stability terms to identify drug degradation, decomposition during storage. So, all these parameters are important to know the formulation basically designing, during formulation designing of any particular type of medicinal product. So, I hope you understand all these basic concepts. So, we will be meeting in next session also regarding the same topic. Till then, take care, stay safe, thank you.